Wonderful. Okay. Well, thank you for that. I'll start over again. I've had a great time here so far. And I'm really impressed by the turnout at 3:36 p.m. on a Friday. It's awesome. So thanks, everybody, for coming. I'm Chris Rhoda, quantitative ecologist at West Virginia University. And when Hemanta invited me, I was like, what would you like me to talk about? And he's like, I think occupancy models would be great. And so I was like, all right, this is an awesome opportunity for me to get out and show my statistical know-how. So especially the undergrads, you guys got to sharpen your pencils. There's going to be lots of equations. But not really. Let's see here. Now the next challenge is to get my slides to advance. I promise I made more than one slide. Okay. Hemanta invited me to come talk about occupancy models, and so I thought long and hard about how to structure a talk that I thought would be interesting to a broad audience. And I decided early on that straight up stats talks are pretty boring, especially at 3:30 on a Friday. So I promised no equations today. What we're going to do instead is I'm going to talk about one of my many research trajectories, but probably my most productive research trajectory, which would be the work that I started as a master's student and have continued as a professor doing development of occupancy models, and in particular multi-species occupancy models. So we'll kind of start at the beginning with some of my work as a master's student. And my first paper published ever is my most cited work as a master's student. So we'll start there with some occupancy modeling work I did. Go through some stuff that we've published just, I think, last year on occupancy models. And we'll talk about some of the fun applications of those models, some of the cool things we've learned from them along the way, and other interesting things that I have learned as a quantitative ecologist. And I should also say that I think almost all of these pictures are pictures that we took of either I got them from my game cameras, or my students got them from their game cameras, or we got them from my pubs. So we do a lot of game camera work in the lab, which is what inspired a lot of the occupancy modeling work. And this is one of the 350,000 pictures that my master's student poured through for her work on white-tailed deer. Okay. So start at the beginning, way back when I was a master's student at the University of Florida. Yeah, this is maybe a little bit confusing. My research was never actually at the institutions where I went to. So when I was in Florida, I did research in Montana and Idaho. When I was in Missouri, I did research in South Dakota. So that was kind of fun. But I did my master's at the University of Florida. My major advisor was Rob Fletcher. I was his first graduate student. And he had me out doing avian point count surveys in Montana and Idaho. So it was actually amazing, right? Florida in the summer is hot, a lot like Texas, but probably a little bit more humid. And so in the summertime, I got to abandon my wife, and I got to go up to Montana and Idaho, where the weather was perfectly delightful. So we were doing avian point count surveys up there. And our sampling methodology was such that we could be fitting these open population models to our occupancy models, right? So we'd go out there and we survey birds for 10 minutes at a time, but we then divide those 10-minute surveys into 4 2.5-minute sampling intervals. Every time we would basically do a brand new point count. And what this allowed us to do was ask if bird communities are open or closed over the course of the breeding season, right? Because usually when we're doing these occupancy models, we're doing it over relatively long periods of time. We're assuming closure over the breeding season, and then using that as a basis for estimating the detection probability and occupancy. And so with the way that we collected data, we were able to ask, are avian communities closed during the breeding season? Okay. Summer in Montana doing the field work, always a strong interest in the quantitative work. And so back in the fall and the winter in Florida, just learning occupancy models. This was the same time I was learning R and all of these analyses, and my advisor really pushed me hard to learn these techniques, and I found them interesting, so it became easy to do. Okay, so from our original question, are songbird communities closed during the summer breeding season? And our analysis strongly suggested no, that they are not. Okay, and to orient you to this figure here, this is a figure from my first pub as a master's student. Basically, if it's got a nice solid bar, that means there's strong evidence that the population is open during the breeding season, right? Meaning that the songbirds, if they're present and singing on a territory early in the season, doesn't mean that they still occupy that territory late in the season, okay? And that if we apply occupancy models that assume closure over the entire period, we might get biased estimates of detection probability and ultimately occupancy probability, okay? So we found strong evidence that a lot of these bird communities were open. In our paper, we developed a test for closure. So folks that were interested in testing whether populations were open or closed could. We also did some substantial simulations looking at the effects of closure. And basically what happens is if we're assuming a population is closed, okay, that second time we go there and they're absent, we're assuming they're present when they're actually not present, okay? So we get deflated estimates of detection probability, which makes us overestimate occupancy probability, okay? And so those are some of the lessons we learned from developing a test to close for occupancy, developing a test for occupancy closure. And to date, this still is my most cited paper, first one I had as a master's student. And that's what got me started into my journey down the world of occupancy modeling. Okay, so we're going to fast forward now to my postdoc. Okay, went to University of Missouri, which is where I got to know Dr. Kafley. We were grad students there together, and did blackback woodpecker stuff for my dissertation. And then working on my postdoc, I started getting collaborative opportunities, doing lots of work with game camera data. So some folks might know of Roland Kays and Wildlife Insights, and a lot of the game camera work that group does. I was able to get involved with that work, with that group. With that group, and with all the game camera data we have, for those of you that have worked with game camera data, you know that you can get detections of lots of species simultaneously. Okay, and I try to illustrate that here with another one of my master students' game camera photos, where we have a coyote and a wild turkey. I asked her not to present this during her master's defense, because then everyone would think that the coyotes were killing all the turkeys. But I can certainly present it here as part of a multi-species occupancy talk to highlight that these game cameras are really effective at modeling lots of species simultaneously. Okay, and so I'm working with Roland and his group, and we're wanting to look at multi-species occupancy modeling, and he's asking me if I can do this. And I'm doing a review of the literature, the multi-species occupancy stuff, and at that time it was really unsatisfying. Right? What Roland wanted to do was count how many people they took pictures of, or count how many coyotes they took pictures of, and use that as a covariate in an occupancy model. But for those that have done much with game camera work or no occupancy modeling, just because you don't detect that coyote on the camera doesn't mean it's not there. And in fact, coyotes have a pretty low detection rate on cameras. And so I found that completely unsatisfying. A lot of the other techniques out there were limited to 2 species. They required asymmetric interactions such that you had a superior competitor and then another species that depended on them. So I found those unsatisfying, and I had a wonderful PhD advisor, postdoc supervisor, Josh Millspa. For my postdoc, he said, you can really do whatever you want to, and I'll pay you, and you can stay for as long as you want. as long as you're like kind of looking for jobs. And so I was like, Josh, I found this really interesting. Can I work on these interesting projects, and can I get my master's in statistics while I'm doing it? And he's like, yeah, go ahead. And so both of these papers that we ended up publishing in 2016 came from my postdoc and came from work that I did while I was getting my master's in statistics. This first one here is, I think this is my favorite of the two, although this is the one that nobody's heard of because I didn't make it all fancy in a package for everybody. But for the first one, I developed a two-species spatial occupancy model. Okay, and so although I applied it to two species because I wanted to keep it tractable for a thesis, it could, in principle, apply to many more species. So what we did in this one is we had lots of these detections of, in this case, we had coyotes and red fox, and we would have a spatial random effect. And that spatial random effect accounted for both the effects of how close cameras were spaced together, but also whether or not one species was more likely to occur in the presence of another species. So we were able to count simultaneously for both spatial and interspecific dependence through those spatial random effects. And these were the results. I pulled this out of my 2016 ecology paper here. And for those of you that haven't been to West Virginia, Morgantown is right here. Little did I know at the time that my future home was going to be in this map I produced here. But this is kind of cool. This is showing how related coyotes and red fox were in space. And so like up in the Northeast in some of those urban areas, we found that they were really more likely to occur together, whereas when we start to get into the southern and western part of the area, they really were more likely to occur apart from each other. So we were able to gain some interesting insights into interspecies interactions. Is everything all right here?
I'm going to work on the Zoom to share the screen.
Okay.
Sorry.
That's all right. Can we go to the next one while you're doing that? There it is. Okay, and I can have this one up in the background while they're working on Zoom. Okay. So that's the paper that turned into my stats thesis, and something we got published in Ecology in 2016. And then approach 2, which is this one here, this was a product of a statistical computation class. We all had to do a big class project at the end. And I had another idea about how we could account for multiple interacting species. This was through a multivariate Bernoulli distribution. All right, so tiny bit of stats in here. A Bernoulli distribution is the distribution of a coin flip, okay? What's the probability of getting ahead, okay? A multivariate Bernoulli distribution is the distribution of lots of coin flips at the same time. What's the probability of getting 2 heads out of 5 tosses, okay? And so I had this idea of drawing on a multivariate Bernoulli distribution. I had no idea if this would work at all, but it ended up working smashingly, okay? And so what we did for the class is I was able to write the likelihood of the multivariate Bernoulli. We applied a detection model on that so that we could account for imperfect detection. And because this was a statistical computing class, I had to write a wicked fancy Gibbs sampler. I think my Northeast heritage is coming out in that sentence there, which got published as an appendix to the paper. And here we're able to do. I'm still really proud of this figure, but this is an example of what we're able to do with this multi-species occupancy model, where on the top here we have the probability of each of our 4 species occurring. We have bobcat, coyote, gray fox, red fox. Coyote keeps coming up over and over again in these models. And we can see, well, how does the probability of one species depend on the probability of the species in the row? And we're able to pull out some really interesting dynamics. Let's see if I can hide this so that people in the room can actually see what's happening here. Okay. Can I minimize this a little bit?
Click on this single dot,
There it is. There we go.
and then if you hit OK at the top, it should get rid of that bar too.
There we are. Okay. Okay. And then here, I think I can even. Oh, now I'm going too wild. I think I'm not going to go that wild. This will be good. This will be good. Was I?
It says it's hide floating controls or something down. Up. There. One more.
You'd think after living through the pandemic, I would know this. But I think that it has just been erased from my mind. Okay. So for example, here we're able to see how coyote presence might depend on the presence or absence of bobcats, right? So when we have little disturbance or relatively wild areas, coyotes are more likely to occur at a site when bobcats are present, and they're less likely to occur at a site when the bobcats are absent. And then as that relationship switches as we have more urbanization. Okay, so we're able to draw really interesting insights into species interactions along ecological gradients with this multi-species occupancy model. Okay. Okay. Oh, and there we are. And so I'd like to highlight some of the cool things that we've been able to do with these models in my lab, and it highlights some of the other research that we do that's not necessarily quantitative, but interesting applications of these quantitative techniques. This here is a little bird that's near and dear to my heart. This is a Canada warbler, and we've worked on this bird for several years now in my lab. This is a bird of conservation concern throughout most of its range. It breeds kind of throughout Canada. And then there's this tiny little distribution that comes down into the northeastern United States and follows the Appalachians. And basically, they're relying on those Appalachians as relatively high elevation habitat that mimics their breeding territory much further north. Okay, but you can imagine climate change, among other things, is potentially causing habitat loss for this species of conservation need. And we've had several research projects in my lab focused on understanding habitat use and demographics of this species. So I had one student, his name was Gordon Dimig. This is actually a picture from another master's student's project, from Stephanie Augustine's project. But we had a student, Gordon Dimig, who was out doing these point count surveys for Canada warblers and all the other breeding birds for a couple of years. And he was collecting multi-species data with these point count surveys, including hermit thrush and black-throated blue warblers. And part of his thesis was to look at habitat use of Canada warblers, and also to look at interspecific interactions to see if they occur more or less often with some potentially competitive species. And what he found was that within our region, the Canada warblers were really closely associated with rhododendron and proximity to riparian areas. And this was a somewhat novel finding, right? Lots of the habitat work had occurred in the boreal forests. And this is a ground nesting species. They like a really shrubby understory. And in lots of the range in Canada, they're actually really associated with forest management and regeneration that occurs after forest management. And we found that not to be the case at all in West Virginia. We actually oversampled recently harvested areas, expecting to find them there. And we found no association with harvest, but we found this really strong association with riparian areas and rhododendron. And interestingly, we found them at the greatest occurrence mid-elevation. Okay, we are expecting to find them at the highest points because of that association along the Appalachians. But we found them at the greatest occurrence mid-elevation. And we found that they were pretty strongly tied with black-throated blue warblers, okay, even after controlling for all those habitat variables. So we think that there might be some conspecific attraction happening here with, say, black-throated blue warblers, okay? our Canada warblers tend to occur kind of in groups. If there's one Canada warbler, there's 1,000 Canada warblers at your site. And you could go a mile down the road, and it looks like it's perfectly suitable habitat, but if there's no Canada warblers, there's nothing. It's not like they occur in low density. They're all or none. And we think that they might be using some conspecific attraction, maybe with black-throated blues or some other species, to find suitable habitat. Okay, so that's some Canada warbler occupancy work we did in my lab. Just to change things up a little bit, one of the wonderful things about being a quantitative ecologist is you can work with everything. So this is the outcome of a collaboration I did with Brock Huntsman, who was a postdoc at WVU at the time, working with brook trout. And he had seen my multi-species occupancy modeling paper and was interested in applying it to brook trout and brown trout. I do have to say this is one of the few pictures that I poached from the web. There's no way I could get that good of a picture of a brook trout. Okay. But part of Brock's postdoc was looking at stream restoration in Appalachia and how it could benefit brook trout. Right, we are really heavily impacted by acid mine drainage in Appalachia, and there's lots of restoration work going on in mountain streams. A lot of it involves dumping limestone just below the source of the acid mine drainage to neutralize it. But there's other stream restoration techniques that we do as well. Okay, and he was interested in asking how does the presence of a non-native competitor, the brown trout, influence the effects of stream restoration for brook trout. This is actually an interesting one as well because brook trout are a species of conservation concern in Appalachia, and I think they're kind of a nuisance out this way, aren't they? Or is that further into the Intermountain West? Not a fisheries crowd. Okay.
No brook trout anywhere close.
No brook. Further west?
They're an issue farther west.
Further west and north, right? In the Rockies. All right. Okay. Let's see here. I love this figure. I think this is a really cool figure. I can claim exactly 0 credit for it. This is all Brock. When he showed this to me on the draft, I was like, Brock, I love it. You got to keep it. Maybe I can take a little bit of credit because it was based off of that multi-species occupancy model that I had developed and worked with him to implement here. Okay. So what Brock found was that habitat restoration was indeed beneficial to brook trout, but only when there was no brown trout in those streams. Okay. He found a pretty strong negative correlation between presence of brook trout and presence of brown trout, which is what we're trying to illustrate here in this figure. Okay, so this dashed line would represent a one-to-one relationship, meaning that brook trout were just as likely to occur at sites with and without brown trout. Okay. One of those implications being that restoration for the brook trout might be most effective at those sites that don't already have brown trout, and that another potential restoration technique would be the removal of the non-native brown trout, which might be out-competing the brook trout in these Appalachian streams. Okay. So there's some interesting applications of our multi-species occupancy model. Up until that point, let's see here, I had that wicked fancy Gibbs sampler that I think exactly 0 people actually ever used. But it got me a stats thesis, so I was satisfied with that. Otherwise, I had provided some, in the original paper, some STAN code. For those of you that don't know what STAN is, it's a software for doing Bayesian modeling. Okay. And so every now and then I would get an email from somebody saying, hey, I found your paper, I found the STAN code, I want to adapt it for my system, how do I do that? And I would help them the best I could, but basically for the longest time, if you wanted to implement this model, you needed to have some skills in Bayesian modeling, which limited the potential pool of users. I was very fortunate to have worked with Ken Kellner. He was a postdoc in my lab for a year. Ken, I think Ken is getting to be pretty famous, especially among grad students. He's got the UBMS package, which allows you to do Bayesian inference with Stan. He wrote JAGS UI, so if anyone in here has ever used JAGS UI, that's how Ken's work. Awesome programmer. I can't believe he ever agreed to come work with me in my lab for a year. Okay. Among the other things that Ken did, and his name will pop up a little bit later here, was he was able to wrap the multi-species occupancy model into unmarked. When he was in my lab, Andy Royal sent me a note. And he's like, hey, we'd really like to implement your model. Can you help us out? And I had exactly 0 time. And so I pawned it off to Ken. And I said, Ken, do you think you could do this? And he took it up and he ran with it. And he did an amazing job of being able to package this in unmarked and make it such that the interface is really familiar to folks that may have used unmarked for other things or may have used R for maybe even just linear models or generalized linear models. Okay, so a really nice, easy-to-use interface. And because of that, use of our model has really picked up substantially. And in fact, one of the lessons I learned from this, especially for you budding quantitative ecologists out there, is if you want people to pick up your techniques, make it user-friendly. Okay. And so let's see here. Earlier on, I said I had 2 of those multi-species occupancy models that were published in 2016 as part of my journey through a stats degree. My second most cited paper is that multi-species occupancy model, and it's like 4.5 times more used than what I think is the coolest model that I wrote, but it's not written in a very user-friendly way. Okay. So if you want broad uptake of your techniques, make it in a nice format, right? That's why Mark Recapture took off, is because Gary White wrote Program Mark. That's why occupancy models and admixture models are so popular, is because they're implemented in unmarked. Okay. So the key to wide uptake is to make your analysis user-friendly. And I've actually applied that in different ways in my lab moving forward now, where when we talk about developing products, we're not just so I've got a new project with the National Park Service, estimate deer density in the northeastern United States. Right, we're not just estimating that density, but we're providing a nice tool that managers can use to estimate density from the data that they're collecting. Okay, so package your analysis, make it user-friendly if you want broad adoption. Right. Okay, we're going to continue down this path a little bit. Before, when people had been using the model, they had to have some kind of a decent statistics background if you're going to implement this in Stan. Okay, it's now an unmarked. Anybody with a laptop and a little bit of coding experience can do it. And so now the tires on this model, we're starting to get kicked pretty darn hard. And we found that there were some shortcomings to the model that we hadn't discovered before this widespread use. Okay, so I got suddenly inundated with these emails. Dr. Rhoda, I got these massive standard errors. Dr. Rhoda, I got these confidence intervals that span 0 to 1. And at first I was just like, well, all you have to do is go Bayes. It's that easy. Just get a weekly informative prior on some of these parameters, and it's gonna solve your problem. And I think exactly 0 people took that advice because the reason they were going to use it in unmarked is because they didn't have the skills to go Bayes. Okay, and so this led me to think much harder about how I could start to solve this problem for the many people that were using it. And here we just have an example of what some of these massive standard errors look like, right? Here's the interaction term between our wood thrush and our Canada warbler. With a standard error of 507, it's completely worthless, okay? So we had to solve this problem, and so that was the next step along this journey, okay? So first I had to figure out what was causing the problem. Why were they getting these massive standard errors? Ken dived deep into the code. There was no problems there with the code. It was written correctly. What it ended up being is what we call a boundary estimate in statistics, all right? So if we never have two species occurring together, or we always have two species occurring together, the model wants to say they're always together or they're never together, okay? And that's why we got that negative 13 with a massive standard error, okay? Because they were never together. But that's something that a multi-species model should be able to handle, right? Two species never occurring together, especially when you start to get bigger and bigger assemblages of species. And I couldn't tell everybody to go Bayes, because especially if I wanted broad adoption, because I think that's outside the wheelhouse of most people. And so I needed something to rein in these extremes, right? In a Bayesian realm, you can have a slightly informative prior distribution. It'll rein in those estimates, and you're good to go. In the frequent twist realm, you need to do something a little bit different, okay? And so I played around with what we call a penalized likelihood. And basically what that would do, it would apply a penalty for those really big values and shrink it back to something that was a little more reasonable. So in my spare time, which when you're an assistant professor trying to get tenure is usually at 10 o'clock at night after the kids are to bed with some beer, I played around with this a little bit, and I found that a penalized likelihood could solve this problem easy. So what I ended up doing, and this is a model that I've been following for a little while now, and it's been pretty successful, is I convened a class of graduate students, and I said, I have a paper that I want to write. I have a proof of concept. I'll give you guys graduate credit and a publication on your CV if you just get it done for me, okay? And this was one of those examples that was led by graduate student Hannah Klipp, and she ended up publishing this in Ecology in 2021 as a statistical note where we were able to apply that penalized likelihood that reined in those unreasonable standard errors. And I tossed Ken a bone. I'm like, hey, Ken, if you can implement this in Unmarked, we'll make you a co-author. He said, deal. Okay. So now in that same function in Unmarked that you can fit the multi-species occupancy model, there's just one little argument in there that says penalty. And you just put a number in there, and it solves all your problems for you. So one little number, the result of a lot of work. Okay. But what it does, right? So on the previous slide, I forgot to mention this. Here is an estimate of wood thrush occurrence with the unpenalized model. Don't know about you, but that looks completely worthless to me. I learned nothing about wood thrush. They might be there or they might not be there. Well, I could have told you that before I started the study. When we apply the penalized likelihood, this is the same wood thrush data. And yeah, at that really low elevation, we still had those really wide confidence intervals. But this allows us to see a much stronger and quadratic relationship with elevation. So we can at least make some meaningful inference from our data, okay? And this technique of actually having proof of concepts and having my grad students follow through, we've been really good with that. We just got a Baer paper published looking at home range. We had another paper that we got picked up for methods in ecology and evolution. If I thought harder about it, we could probably find some more. But this has been a great way to get grad students engaging with quantitative work and getting some nice side projects out. Okay. So there we are, penalized likelihood. Okay, this is another paper that I can't take any credit for. We published the penalized likelihood, but then I've been helping graduate students at WVU apply this, and so give this talk next year, I'll probably be able to have one of their papers up here. But this was a nice application I found by following citations of Hannah's paper that has to do with evaluating the human shield hypothesis. So this human shield hypothesis is an interesting idea. It's the idea that some prey species will actually use humans as shields against predators. Okay? And the idea is that some prey species will be more abundant in close proximity to humans because they have a lower chance of getting eaten. Okay? And this came out of Olivier Jimenez's lab. So he's a quantitative ecologist in France, and when I saw him pick this up, I'm like, yes, because he's actually quite influential in his own right as well. And there's lots of interesting things to pull out of this, but his evidence of the human shield largely comes from the lower right-hand panel here, all right, where we've got occupancy probability of prey on the y-axis, conditional on the presence of humans. And on the lower left-hand side here, we have low forest cover, and here we have high forest cover. Okay? And so we see that those prey are more likely to occur in areas of low forest cover when people are present relative to when people are absent. And so this was a really interesting application of that penalized likelihood. And it was also cool to see that basic template for my figure that I developed way long ago replicated again. Okay. So folks able to use that penalized likelihood to draw some inference on human shield hypothesis. Okay. Okay. And now we're going to switch gears a little bit. We're going to keep talking about multi-species occupancy models, but we're going to talk about some of the more recent developments that we've been able to make in my lab. And actually, this development had roots that were quite a bit older. Roland Kays, I had mentioned his name a little bit earlier. He had invited me to come and do a workshop on multi-species occupancy at a conference in Perth, Western Australia. Great time, although that's what gave me my anxiety about getting the flights way early. That's another story that I'll tell you over beers after the talk. Big mammal workshop in Perth, and as you do, we were sitting around drinking, talking about game camera data and analysis. And Roland's the big thinker kind of guy. And he was like, what I really want to be able to do is ask how the presence of one species affects not necessarily the presence of another, but how it affects activity patterns of another species. How can we do that? And again, this was, the answer to this was unsatisfying to me as it currently existed. Because they usually just involved ignoring imperfect detection of the other species and correlating temporal detections of one species with another. Okay, so I had some ideas. In my hotel room, I downloaded a couple of papers on point process models. But if anyone, we were talking about Australia today. Somebody had been leading an education abroad trip to Australia. It's a long freaking flight. It was like 14 hours from Hong Kong to Boston. So what do you do on a long flight like that? Well, you do a little bit of programming. And so by the time I got home, I had been able to implement a point process detection model. We were able to look at how presence of one species influenced activity patterns of another species. And this is what came to be known as our continuous time multi-species occupancy model. And Ken comes into play yet again on this one. I wrote this proof of concept up back in 2017, I think is when that conference was. And then this is another one of those things where it wasn't funded, and I couldn't dedicate a lot of time to it. So I said, Ken, I got this idea. do you think that you can code this up in a user-friendly way, which Ken is amazing at, and get this published? We were able to publish this in the Journal of Agricultural, Biological, and Environmental Statistics. It came out a couple of years ago because it was one of those unfunded projects. But what this allows us to do, among other things, is on this figure here, we're looking at now not probability of occurrence, but temporal activity patterns of white-tailed deer, which is the solid line, in the presence and absence of coyote. Okay, so on the top there we have white-tailed deer when coyotes are present, and on the bottom here we have white-tailed deer when coyotes are absent. Okay, and what we see from this is that there's a lot more white-tailed deer activity. There's a lot more whitetails detected on cameras at those sites where there are coyotes. And there's a slight shift in activity patterns as well. So their diurnal behavior isn't as pronounced when coyotes are present. And their activity is much more evenly distributed throughout the daylight hours. Okay? So this allowed us to address questions in a different way, right? It's one thing a species can affect the other spatially, but it's actually really important to understand how they might affect other species temporally as well. And we learned that from COVID as well, right? As humans, space use changed across the landscape during COVID. We saw changes in species, not just in space, but also in time as well. And those are dynamics that this model allows us to get at. And so this is an application of that model in practice. Okay, so for the paper we published in JABES, that was just a small test data set, proof of concept. Yeah, this can work, and it provides interesting results. And this is actually a side project from my lab as well, along the lines of the penalized likelihood paper. Okay. The students were interested in asking how predators influence activity of prey. We again used game camera data, but a different set of game camera data than we were using for the proof of concept. Okay, this is game camera data mostly in West Virginia and a little bit up into Pennsylvania. The broad question was, how do predators influence activity of prey? And just because white-tailed deer are the most abundant animal by far on all of our game cameras, and coyotes are probably really their only big non-human predator on the landscape in that area, we picked coyotes and white-tailed deer. Okay? And interestingly, we found the exact same thing as we found with that test data set from JABES. That presence of coyotes on the landscape really influences activity patterns of white-tailed deer. Not only did that, but we were able to catch in this sequence here a really cool chase scene. So if you look, these pictures were all taken within a minute of each other. They all have an 11:03 timestamp. And there goes the white-tailed deer, and there goes the coyote chasing the white-tailed deer. So we were able to capture those dynamics in our model, but we were also able to get a picture of it as well to prove to the doubting peer reviewer that coyotes in West Virginia actually will chase white-tailed deer. Okay. Geez, this paper was just published in like last month, I think it was. We just got this paper out, and so it gets us right on time here. So this is a talk of my journey through occupancy models, starting as a master's student and playing with a test for closure and evaluating what happens to our inference if we violate closure, through development of several multi-species occupancy models. The lesson that if you want people to pick up your model, make it available, all the way down to development of the continuous time multi-species occupancy model, and cool things we've learned along the way. With that, I am happy to take any questions. This is also a picture that my grad student Sarah took for her whitetail project as well. She just had the best game camera pictures. But with that, I'm happy to take any questions. In terms of predators, they're probably the most, it's hard to say. Black bears in the area might be tied approximately with coyotes. But in part, I think it's just because they're relatively low density and they have relatively large home ranges.
Do you think that they can smell the human scents on the camera?
Man, yeah, I do. Coyotes have really good sense of smell. I know the animals know the game cameras are there. You can tell it with black bears because they will actually eat the camera. But I've got pictures of coyotes where you can tell that they're coming up close to the camera. They know something's wrong, and they hightail it the other way. And then they just know. I spent a lot of time sitting in a tree stand trying to put an arrow in white-tailed deer, and I couldn't tell you how many coyotes I've seen. They're just trotting along, and then they just know I'm there. They look at me, and they're gone. So, coyotes know those cameras are out there. White-tailed deer know those cameras are out there. Yeah, I think all the wildlife does. It's not as stealthy as we'd like to think it is.
How do you come up with numerical equivalents for activity or detection? So is it a longer period of activity will be more numerically significant, or how do you track that?
Yeah. So how do you come up with how do you quantify activity patterns?
Yeah.
Okay. Let's see if I can get into this without getting into too nerdy of technical details.
I can give you marker.
Nobody wants to see me write out the likelihood of a Poisson point process. So the detection model there is it's a Poisson point process model, the timestamp itself is the quantification of activity, and then you can think of that likelihood in several different ways. If you want to think of it as a Poisson process, you might have some really small chunk of time, and then you could think about the number of detections you would expect in that small chunk of time. But then a Poisson distribution and an exponential distribution are actually closely related. An exponential distribution is the waiting time between two events that might occur in time. And so you can also take that point process and divide it among temporal intervals like that. So there's lots of different ways that you can cut up that likelihood. But the way that I'm using it is just as a timestamp and thinking about how many of those discrete events might occur in a small period of time.
You spoke a lot about coding for your models. How great of a knowledge do you have of geographical informational systems, and do they help you with your modeling?
I use GIS stuff all the time. You may have seen it in that map that had West Virginia and stuff in it. So the short answer is yes. Knowing how to use GIS can really help with this modeling here in so many different ways. One of the biggest ways I might do it is to obtain remotely sensed covariate layers that we can use as predictor variables. But even basic mapping like this. So one of the things that I really try to push my students to do is develop pretty strong data visualization skills, and GIS is really handy in those data visualization skills as well. And there's lots of other reasons that it's handy.
So would you consider it one of your first steps before actually forming the models with the coding stuff?
No, it's not one of my first steps. I'd say that it comes along the way. I kind of use it as a, well, tool for covariates, but you got to have the model first if you're going to use covariates, or if I use it for mapping, that comes kind of after the fact. Yep, so it's definitely a part of, it's almost never the first thing I do. Yep. Other people might give you a different answer though. Yeah, I say just because I do it this way doesn't mean that's the only way it has to be done. Yeah.
What software do y'all use, or if y'all use any at all, for going through your game camera footage?
Wildlife Insights. Almost, I think we exclusively use Wildlife Insights. And I think that's just a natural accident of me having fallen into Roland Kay's group, and the eMammal was like the predecessor to that. And I was involved early on in helping with eMammal, and then that morphed into Wildlife Insights. It's just a really, I promise they're not paying me anything, all right? I find it to be really easy to upload your data. It automatically sorts your data into sequences, spits out a nice spreadsheet for you. And the AI, I won't lie, it's not awesome. But Wildlife Insights does use AI to try to tag animals, spot on with humans, spot on with what I call blanks. So if there's nothing in there, it's really good with that. It often calls my white-tailed deer mule deer, and there are no mule deer in West Virginia. But if it can get it to like Artiodactyla, that's good enough, because the only thing that could be in that region is a white-tailed deer. It gets raccoons really good if it can get their tail. That's what I used. There might be better platforms out there. I've just never needed to explore, because Wildlife Insights has always suited my needs. Yeah.
Thank you for a very interesting presentation. I just have a question about how many factors can you include at a time in your model, and how certain are you about some of those cause-effect relationships? So I can see that the coyote and the white-tailed deer interaction, it's very nicely illustrated. But what about factors like bush cover, pasture cover,
Yeah.
seasonal changes, and so forth?
Okay. So 2 questions. Don't let me escape without answering them both. First is, how many variables can I have in the model? I'll give you my favorite answer, and it depends. A rule of thumb that I try to stick to is try to keep 20 or so observations per covariate. It's based on a really loose rule of thumb that I read in a paper that was written by Ben Volker, so I trust it and I cite it. There's nothing magical about that. It's just kind of a rule of thumb. So if you have really sparse data, I don't really ever let my students put in a lot of variables to keep the model parsimonious. So I think that's the furthest I'm willing to answer your first question, unless there is something else you were looking for.
No, that's good thinking.
Yeah. For the second one, what I didn't mention here is we did include NDVI, normalized difference vegetation index. It's a measure of greenness as a covariate, and it actually came out to be an important predictor of activity as well. It didn't change the overall relationship between coyote and white-tailed deer. And I think what's important to keep in the back of our minds, and maybe I should have led the talk with this, is pattern doesn't equal process. So we're getting lots of correlations here. And so maybe there's something that's going on between the presence of coyotes that's causing greater activity of white-tailed deer. But beyond that, it's pure speculation, right? And these models might not be the best suited to address what's the mechanism behind why white-tailed deer activity increases when coyotes are present. Yeah.
Any other questions?
Chris Iyer.
Yes.
I know it's coming.
So going back to the penalized likelihood and those standard errors, which I hope the students in my stats class recognize that before you had pointed out. But was that mostly an artifact of the data set, or if you have what if you have more evenly distribution of detections and absences? Are you still going to have to implement the penalized likelihood?
No, you don't always have to.
Okay.
Yep.
Does the
No, if you apply it and you don't need it, it will do something.
Yeah, okay.
Yeah. But you'll know if you need it because you'll get those outrageous figures like I showed up
The
there.
standard errors?
Yeah. There's no question if you need the penalized likelihood.
Okay, cool.
Yeah.
Thank you.
You're welcome. That was easy. I was waiting for something hard.
I don't like to do that to people in front of a bunch of people, so well, I'll do that in private.
Okay. All right. All right.
Any other questions? It's incredibly informative talk. Thank you so much.
Yeah. Thank you.